Substances are absorbed in the small intestine, the broken down food molecules, and this happens in different ways, and I'm going to explain this right now. What you see over here is the phospholipid bilayer of a cell which makes up the villus, which absorbs the food molecules. And there is a separate video relating to the structure of membranes. This is not our main focus right now, so you might want to watch that video first if you do not understand what these little things are over here. These are phospholipids. So, but we now do the following. Let's say that this is inside the cell, and this is in the lumen. And the digested food molecules are over here. And in the case of simple diffusion, the food molecules are able to squeeze between the phospholipid bilayer and they're able to diffuse to the inside of the cell. Look how I spelled cell over here. I forgot an L. Okay, so, but what you need over here is you need a high concentration of food molecules because you need the concentration gradient. Now the inside over here, the inside of the phospholipid bilayer is hydrophobic, and therefore only substances which are also hydrophobic are able to go through. But that is basically a form of simple diffusion, nothing special about this. More interesting is this facilitated diffusion. It is still diffusion, therefore you still need a concentration gradient over here. I'm not going to label lumen now in the inside of the cell. It's the same. So we have over here now a high concentration gradient, but now the issue is that these molecules are hydrophilic. They're water soluble, and therefore they don't want to do that. They don't want to go through here. They can't, because the inside here is hydrophobic and the food molecules are hydrophilic. So, but what you have over here is you have a so-called channel protein. And this channel protein now has a little hole in the center, and these hydrophilic molecules are now able to go through to the other side by diffusion, because the channel protein makes it easier to diffuse. It's called facilitated diffusion. Does not require energy, just same as simple diffusion. You do not need ATP or any other energy source. Examples for substances that do that, for example, is fructose, which is fruit sugar. Now, in some cases, we do not have this concentration gradient, or we would like to have a higher concentration inside. And in this case, we need active transport. Again, we have the outside of the cell over here. And now these are minerals. And they have to be enriched on the inside. But the problem is over here is that the concentration in here is higher than on the outside. So they do not want to voluntarily move in. So, but what's going to happen is the following: is one of these minerals is going to go into this protein pump. This is a pump, and it's going to flip over. But this requires energy in the form of ATP, and this ATP is broken down to ADP plus an inorganic phosphate, and this process releases energy. And now we have the mineral, which was on the outside first, now on the inside, and here we still have a high concentration of minerals, so we're able to pump the minerals against a concentration gradient or up a concentration gradient, and as a consequence we will have a higher concentration on the inside than on the outside. In the case of simple and facilitated diffusion, which I just talked about, essentially you will have then an equal concentration on both sides. But over here with active transport, we really start to build up here a high concentration of minerals. Next one is called endocytosis. And in endocytosis, we see that the membrane itself folds in. So if this is our cell, the gray one here, that's our nucleus. And if you have over here, let's say fat droplets, these are the so-called triglycerides or certain lipoproteins, what will happen is that the membrane will fold around them and it will start to make a so-called a vesicle. And this vesicle contains now these small food molecules, because they cannot be taken up directly across the membrane. This process is called engulfing. But the better term that you would use is probably endocytosis. I say engulfing because it is similar to what phagocytes do when they take up pathogens. But the idea is the same. There's an infolding. It's an infolding of the membrane. Now there are also other more complex uptake possibilities, and it's so-called the sodium co-transporter. If this is the outside of the cell, and this one is the inside, what we do is we have a combination between active transport of sodium. So sodium is pumped out of the cell, Na plus is pumped out of the cell by active transport. So this requires ATP. Okay, you got to add ATP here, and you have a higher sodium concentration out here now than on the inside. Now, on the outside here, we also have glucose, and I'm going to draw the glucose here like a dot. That's my glucose. And the glucose should be taken up into the cell. Now, how can this work? The glucose has to be pumped in, but we will use the energy by the inflow of the sodium. So as the sodium diffuses in, the glucose is taken along. So we have Na plus over here and the glucose over here. And this Na plus then, of course, is again actively pumped out of the cell. And this is called the co-transporter because sodium and glucose are transported through the membrane at the same time. So that's basically was an overview over the different ways of how we can get substances into the cell for absorption.